Hi, good afternoon, everyone, and thank you for joining us for our latest installment of the HGSA Research Webinar Series. Today we have Dr. Daniel Wilton from Children's Hospital at Harvard presenting. Before I turn it over to Daniel, I just wanted to give a few notes about how to write questions. So for those of you who have not attended a research webinar in the past, it's really easy to ask a question. Just go into the questions box highlighted here in the red bar, or red box. Type your question at any time to us, and at the end of Daniel's presentation, we'll make sure that we ask them today. You can just click send, and like I said, anytime during the presentation. And other webinars in the future, you can always go to hgsa.org, and we have something new in the past few months where you can click on, shown here in red, go to our YouTube channel, the icon. You can view this recording at any time in the future. Upcoming in April, we have a webinar, not yet scheduled. Sorry, there's a little bit of noise here. Everyone, if anyone's on, their phone. Sorry. Okay. Hopefully, everybody can hear me. A lot of feedback. In and out. Okay. I apologize. I'm getting a lot of audio feedback from folks telling me that. What I was going to say is that on April, at a date, Dr. Alice Bada from UC San Diego will be joining us to present his recent work from his lab on a KD3010 as a potential therapeutic strategy for the Daniel?
Yes, I can just hear you.
Are you using your microphone attached to your computer or your phone?
I'm using the microphone attached to the computer.
Sounded fine earlier.
It's loud.
Our landline. Bear with us. Is currently a postdoctoral fellow at the department around.
Yes, so you're still getting the humming. Okay.
And
That is
Daniel,
different.
fellow at the laboratory.
That's for
that. Daniel, bachelor's degree at Birmingham.
Yeah, I was like, what is that?
Daniel's going to be talking to to us about a really novel topic in HD and really trying to address the question of does systems play a role in HD biology? The title of Daniel's presentation is The Complement in Synaptic Loss in Driving Disease Progression. Daniel, I'm going to turn it over to you. And
Thank you, George.
hopefully,
Thank you for that introduction.
I apologize for all the feedback. Hopefully everything will be quiet now.
So I'm very happy to be able to present some of my work to you today. So today I'm going to talk to you about the research I've been carrying out in BEST lab, trying to understand mechanisms that drive Huntington's disease pathology. Before I show you some of my unpublished preliminary data, I want to explain some of the concepts underlying my work. So,
Daniel, sorry, Daniel, can you share your screen with the audience, please?
Is it better?
Fantastic. Thank you.
Sure. Okay, so as you all know, Huntington's disease is a neurodegenerative disease, and it affects specific brain regions. The brain itself is made up of many types of specialized cells. One type of cell in the brain are the neurons, which are pictured here, which receive information from many sources and communicate this information to other parts of the brain to coordinate appropriate responses. Neurons are connected to structures called synapses, which I'm highlighting here, and which is shown blown up here, which allow them to communicate information from cell to cell. There are trillions of these connections in the human brain, and they're essential for the correct functioning of brain cells. They also play a key role in memory formation. Synapse loss, or the loss of these connections between brain cells, is something that occurs during normal aging. However, in many neurodegenerative diseases, such as Alzheimer's disease, synapse loss is thought to be one of the earliest events to occur. And here you can see this in this image of a neuron, where these protrusions, which I'm highlighting here on the processes of the neuron, indicate synaptic connections. And in the tissue from the AD model, there are many fewer of these connections than there are in the control tissue. However, the factors that trigger synapse loss in the aged and diseased brain remain poorly understood. What makes these synapses vulnerable? And to answer this question, we've been carrying out studies in the developing mouse brain. So the brain starts out with many more synaptic connections than are actually needed. And this is shown here in the staining of human cerebral cortex, where you can see there are many projections from connecting neurons to one another that are gradually lost as we age. So there are many fewer of these in the sample from the 14-year-old individual. And during development, some of the connections onto the neuron, onto individual neurons, are removed, while others are strengthened and maintained. And this developmental synapse loss is part of a process called remodeling that creates the precise circuits found in the adult brain. A major question in the field and in our lab is how are these specific synapses removed? If we could understand how synapses are normally removed or lost during development, this may provide important insight into how they are lost in these devastating diseases and may lead to new therapies. Our lab has started to understand some of the mechanisms involved, but in order to tell you about them, I have to introduce another major cell type in the brain. These are the glia cells. So there are many different types of glia cells in the brain, and in this picture shows the different types, the oligodendrocytes, which wrap myelin sheaths around axons, the astrocytes, and the microglia. And emerging evidence implicates these cells as having important roles in creating synapses, supporting their function. We now know that glia actively communicate with neurons and one another to regulate regulate synapse development and function. So the microglia are very similar cells to cells actually found in the immune system. In the brain, they respond to injury and disease, clearing away debris and dead cells. But more recently, it's been shown that they may also have a variety of roles in maintaining normal brain function. Most studies of microglia have been performed in the injured or diseased brain. Following injury or disease, microglia are rapidly recruited to sites of damage or pathology, where they phagocytose debris as well as unwanted and dying cells. And you can see here in this movie where the microglia is labeled in green and the dying cell is labeled in red, that as the cell dies, parts of the cell are gradually taken up and ingested by the microglia. This phagocytosis is important for removing potentially harmful material. However, microglia function in the healthy brain has not been well understood. What are microglia doing in the developing brain when these circuits are wiring up? Well, it's been observed that during periods of synapse remodeling, microglia are actually contacting synapses, and this is shown here in this image in blue and red, with the microglia shown in green, leading us to hypothesize that they could be actively involved in moving inappropriate synapses through phagocytosis. To test this, Dori Schaefer in our lab looked in a part of the brain in which remodeling has been frequently studied and used dyes to mark synapses and microglia. She then used microscopes to see if microglia and synapses were interacting. And indeed, she could see synapses inside the microglia. So you can see here this image she's generated where the microglia is shown in green and is made translucent. And you can see synapse components inside the body of the microglia, suggesting they have been phagocytosed or eaten by the microglia. But how do microglia know which synapses to remove and which to leave and to form these precise circuits? Well, microglia are very similar to immune cells called macrophages. Therefore, we used information about how these cells phagocytose for clues as to how microglia might be functioning. Macrophages remove pathogens and dying cells by responding to complement proteins. Complement proteins can be called eat me signals, and here is a picture of the complement proteins. They bind to the surface of cells that need to be removed, such as bacteria, and then macrophages come in and they express receptors to these complement proteins, and they will come and engulf them and remove them from the body. When we stained brains for these complement proteins, we found that surprisingly they were also present in the brain, and they were binding to developing synapses to promote phagocytosis by microglia. So our working model is that microglia removes synapses in the developing brain by phagocytosis, and this involves complement proteins binding to the developing synapses, which are then recognized by receptors on the microglia, which come in and phagocytose those synapses which need to be removed. However, in the healthy brain, this process is no longer active as precise circuits have formed. A major hallmark of neurodegenerative diseases is synapse loss, which leads to the question, could synapse loss in Huntington's disease be caused by an inappropriate reactivation of this mechanism in the mature brain, whereby complement proteins are localized inappropriately to synaptic connections, which are then subsequently engulfed by microglia. in support of this, in Huntington's disease, as in many types of neurodegenerative disease, there is evidence of increased complement proteins in the tissue, postmortem tissue of Huntington's disease patients, as shown here. And we also see, it's also been shown that there is increased activity of glial cells in HD patients, with shown here in this PET scan. So my work is to try and address the question of whether microglia and complement play a role in synapse loss in Huntington's disease, has basically focused on using HD mouse models. And these models are not perfect by any means, but they replicate some aspects of early Huntington's disease. So the first questions we wanted to address with these models are synapses lost in these, and how early does this occur? So to assess this, we stained mouse brain tissue with antibodies to identify synapses. Here, shown here, an overlap between red and green spots indicates a synapse. And you can see that there are many fewer of these colocalized spots in the HD brain tissue. We counted the number of synapses at different ages and found that synapse loss was actually increased with disease progression. And also we found that synapse loss occurred before the onset of disease symptoms in these mice. So these mice only show motor and cognitive deficits, begin to show motor and cognitive deficits at 7 months. But we could already see some evidence of synapse loss at 3 months. So then we wanted to ask the question, are levels of complement proteins increased? And do they localize to synapses in these mice? So to do this, we stain mouse brain tissue sections with antibodies to complement proteins. And there's these, as you can see here in these images, what we saw was that there are much higher levels of the complement protein C3 in disease-affected regions in this model. So in the striatum and in the motor cortex, we could see much higher levels of C3 than we could in an unaffected littermate of these mice. And interestingly, we couldn't see this increase in other brain regions that are less affected in Huntington's disease. It also this this high level of C3 also seemed to increase with disease progression. So then we wanted to see whether these complement proteins were actually colocalized synapses. And to do this, we used very high-resolution microscopy. And what we could see was this was indeed the case. So here, the synapses are labeled in red, and the complement proteins are labeled in green. And in this blow-up here, you can see that the two markers are actually localizing together, suggesting that the complement proteins are indeed being deposited onto these synapses which are lost during the disease. So everything I've shown you so far is data from these models, but we've now established a collaboration with Richard Fall in New Zealand, who operates one of the world's largest brain banks of HD tissue, to see whether there's any evidence of this in human HD pathology. We've obtained tissue at different stages of disease progression, and the staining I'm showing you here was carried out on von Stauder grade 2 tissue, which correlates with a mild to moderate clinical score. And what you can see is that in a disease-affected brain region, the caudate nucleus, there is an increase in the level of C3, complement protein C3 deposition. And as in the mouse tissue, we don't see this increase in other less disease-affected regions. Again, using high-resolution microscopy, we can actually see in this human tissue that the complement proteins were localizing to the synapses, and this is shown again here in this blow-up. So what I've shown you so far is that in the models that we've been studying, there is synapse loss, which occurs before the onset of disease symptoms, and it correlates with an increase in levels of complement proteins which are deposited on to synapses. In development, microglia remove excess synapses through complement, so do microglia also contribute to synapse loss in Huntington's disease? So to see if microglia were phagocytosing synapses in the HD mouse models, we labeled synapses in microglia using similar methods to those used in our brain development study. So here you can see the microglia is shown in red and the synapses are shown in green. And what you can see is that there are more of these green synapses inside the body of the microglia from the HD mice than those from the microglia from a control litamate, suggesting, and when we quantified this, we found that indeed these microglia from the HD mice were actually phagocytosing more of these synapses than their age-matched controls. So taken together, all of these results strongly suggest that synapse loss in these mice may be driven by an inappropriate increase in complement and microglia phagocytosis. So then we will see whether we could actually target complement or microglia to reduce the loss of these synapses. So we took two different approaches to this. One was to target the microglia specifically by removing complement receptors. And the other was to try and prevent complement deposition onto the synapses. And the first experiment I'm going to tell you about was where we removed the complement receptor from the microglia. So we have mice in which the gene for the complement receptor is removed from the microglia specifically, and we cross these to our HD mice to generate HD mice which lack the complement receptor in the microglia. And then we quantified the number of synapses in these mice at a time when we knew synapse loss was occurring. And what we found was that there were more synapses in these mice than HD mice with expressing the with microglia expressing the complement receptor, suggesting that removing the ability of the microglia to recognize the complement that had been deposited had actually helped to reduce the losses in this model. And then the other strategy we've tried is to block complement deposition onto the synapses. So to do this, we entered into a collaboration with a pharmaceutical company called Annexon, who are developing novel blocking antibodies to prevent the binding of complement proteins to synapses. So this would essentially block the deposition of these complement components onto the synapse, and then thus prevent the microglia from coming in and engulfing them. So this blocking antibody, when we treated the mice with this antibody, we found that and then quantified the number of synapses, we found that mice treated with this antibody, relative to those treated with a control, there are significantly more synapses in the antibody-treated mice, suggesting that blocking C3 binding may prevent at least some of the synapse loss normally seen in this model. And in these pictures, again, the green and the red colocalization indicates a synapse, and you can see there are many more of these present in the tissue that, from the mouse that's been treated with the C1q blocking antibody. In collaboration with a group at UCLA, we're now planning to carry out a study to assess whether treatment with this antibody can slow or prevent disease progression in these mice. In conclusion, our preliminary data so far suggests that synapse loss in HD mouse models could be caused by an inappropriate reactivation of developmental mechanisms in which complement proteins are localized to synapses, which are subsequently phagocytosed by microglia. And it may be possible, and it seems like it's possible to prevent this process by either removing complement receptors from microglia or preventing this complement deposition. However, there are many unanswered questions, including what signals trigger early increases in complement in Huntington's disease, and whether reducing synapse loss actually slows or prevents disease progression. I'd like to finish there and thank you for listening to my talk today. I'd also like to acknowledge the other members of my lab, my mentor, Beth Stevens, a talented technician in the lab, Molly Heller, who's been working with me on this project, a summer student, Andrea Kim, who worked a lot on the staining of the human tissue, and all the other members of the lab, and our collaborators at UCLA, William Yang and Anthony Daggett. I'm going to transfer the control back to George.
Great, thank you, Daniel. That was fantastic, really great, and I felt very clearly documented overview of the complement system. So it's a super exciting and, as I said, novel approach that many of you may have not have heard about, dialing in and being involved in HD, and we're very pleased to be supporting Daniel and Dr. Stevens by helping to support some of their research. I forgot to mention, with all the chaos of the audio issues at the beginning, that Daniel is a recipient of the HDSA Human Biology Project Fellowship for the past 2 years to help pursue, to translate some of the findings that he presented today that he saw in the HD mouse model to see if what they're seeing in mice is actually happening in humans. So it's really exciting preliminary work, and we look forward to seeing how this all transpires in the future. If anyone has questions, certainly do ask them now. One question I had was, you presented some work on the C3 knockout, a genetic cross to an HD mouse, and showed some benefit. Have you tried other complement knockout crosses, such as C1q to HD animals?
Yes, so this is ongoing work that we're carrying out in collaboration with William Yang and Anthony Daggett at UCLA, where we're going to look at both the C3 knockout and the C1q knockout in a longitudinal study where we of HD mice, where we look and see whether the progression of the disease is altered in any way by removing these complement proteins.
Does mentioned a C1q antibody with an exon. Does a C3 antibody exist? Is that a reagent that you guys have available to also test in HD animals, or eventually, hopefully, in people?
It doesn't exist at the moment. It would be interesting to look at that as well down the line, and I know that Annexon are working on developing other blocking antibodies, so maybe this will be another target we can address in the future.
A few questions are coming in for you, Daniel. One is, you suggested that the synapse was found inside the microglia with some of your images, the green dots inside the red microglia, if I remember correctly. How do you know that that's truly inside the cell, or as opposed to just maybe stuck on the outside extracellular membrane?
Right. So the way that we measure this is that we image through the entire volume of the cell, and then we use special software to create a 3D representation of the cell. And this software can then be used to only show those synapses which lie within this three-dimensional reconstruction of the microglia. So we're very confident that when we're actually measuring the amount of this material inside the cell, we're really measuring material that's inside the cell and not just material that's on the surface.
Great. Another one for you, Daniel. This is first a comment. It says that normally is complement deposited on all synapses but shed from active synapses, or is deposited only on synapses that should be eliminated?
So that's a question we're trying to address. So some of the work I didn't show today has suggests that complement proteins are localized to specific populations of synapses in the HD mice, and these are the synapses which are lost early in the disease. So it suggests it's a targeted event, but the mechanism which targets the complement proteins to these specific synapses is not well understood, and that's going to be the subject of my future work.
Another part of this question was does mutant Huntington inappropriately enhance the marking of synapses? And then my kind of follow-up to that would be, the community is very excited about the ongoing genetic silencing of Huntington trials that have begun, and all of the preliminary work that's gone on in mice and non-human primate models testing these different like antisense oligonucleotides. Do you have any experience showing that your complement markers decrease if you lower Huntington in your mouse models?
Yes. So we have begun studies with, in collaboration with William Yang's group, in which we have used mice where the mutant Huntington protein is selectively removed from different neuronal populations, and I've assessed complement deposition and microglial engulfment in these mice. And what I see is that in the absence of mutant Huntington expression in different brain regions, I don't see this complement deposition, microglial engulfment, or synapse loss.
Great, thank you, Daniel. Another question is coming from, they wanted you to elaborate on, you mentioned the genetic removal of, I believe it was C3. The question coming from the audience is, can you kind of elaborate how you do a genetic removal of a complement protein or any protein?
Right. So a genetic removal basically, in this case, involves you targeting a specific gene in the embryo itself, in the germline, and all of the cells then in the mouse will no longer express the gene. There are many different ways in which this can be achieved. It's also possible to, using different approaches now, to remove genes from specific cell types, or only remove them at certain time points. It's a complex process. I hope I addressed at least some of that the question you asked.
Yeah, thank you, Daniel. Yeah, and just say to the caller, it's just genetic removal or knockout can be done many different ways. It's just a really nice, it's kind of a tool that researchers use in the lab either to, it's not to the point where this can be done in humans, but in mice and lower organisms, it's much easier to do where we can kind of knock out or prevent the expression of a certain gene. Daniel's made it sound very, easy. They would just simply made a knockout and crossed it to an HC animal and voila. But it's quite elaborate and but done routinely. Can you let's see? There's a couple more questions here. One question. They just wanted to know, how was the complement antibody that you used delivered to the animal?
So,
route of administration.
So this antibody was delivered peripherally. So I injected the mice into their intraperitoneally, which is into their abdomen, and I gave them daily. They received injections every 48 hours for 1 month before I sacrificed them and assessed their synapse numbers.
And I'm not sure if and this might be something that you've already touched on that you're trying to address, maybe no one knows, but exactly, do you know what makes the microglia attack or consume the synapses? I mean, I know it's part of normal development, and then it just stops as you age, hopefully. Become a mature adult, you have the appropriate connections in your neurons. But is there anything else that you think is making microglia attack the synapses in HD?
So in development, this process seems to be linked to neuronal activity. So connections which are strengthened by increasing activity within a particular circuit are protected from complement deposition and microglial engulfment. It may be that neuronal activity or aberrant neural activity also plays a role in targeting complement to specific synaptic populations in Huntington's disease. And that's something else I want to explore in my future research.
Great. One question came in. Do you see genetic removal being done in humans in the future and past the embryonic stage? I mean, I think I can answer that for Daniel, and you can chime in, but I don't see, if we could click our fingers and we could have genetic removal of any of the complement proteins, that's not going to be a viable approach for Huntington's disease. As Daniel said, I mean, this is serving a very important process in our brain development. We need complement proteins, so to knock that out early would probably be very, very detrimental to a human being. So I don't see that as a path forward, but these antibodies that Daniel's working with could be a way to intervene to dampen the complement response in adults. Does that sound fair to you, Daniel?
Yes, that sounds fair.
Do you know anything about the antibody being able to cross the blood-brain barrier?
So we're currently carrying out studies to confirm that the antibody definitely is crossing the blood-brain barrier. We can obviously see effects on synapse numbers, but it could still be that we're having some effect on the periphery, which leads to some secondary effect in the brain. So we're working with an exon to address that issue.
A question. This was involving human trials? Have you considered human trials? I think that's maybe beyond the scope of Daniel's work, but the company Annexon that you've heard Daniel reference is considering these types of approaches, human or clinical trials for this type of approach to dampen the complement system. And in fact, I can say that Dr. Mario Saltarelli, who's the Chief Medical Officer of Annexon Biosciences, will actually be joining us at the HDSA annual convention in Baltimore in June to present some more details on this work and plans that Annexon has for potentially looking at HD as an indication with their antibodies. More question, is the quote-unquote deposit that is being attacked by the microglia, is it the HD protein or a different protein?
So my work so far suggests that the microglia are responding to complement that is deposited onto these vulnerable synapses, and that if you prevent the complement deposition or prevent the microglia from recognizing this deposited complement, then that can protect these synapses. This process seems to be driven by mutant Huntington, but the mechanism through which mutant Huntington leads to complement deposition is at the moment not known.
Great, thank you. Do you think this is also kind of hard, but I'm interested to see maybe you've got some data with the mice or with the mice treated with the antibody, can reversal of the disease be achieved through this process, but through dampening the complement system, like in the HD animals? Is there a point that it's too late to treat them with the antibody, or could you recover function and synapses at any age along the disease progression of that particular animal that you showed?
It's an excellent question, and it's what we're planning to do in follow-up experiments in collaboration with William Yang's group. We don't know at this time whether there's a point at which whereby if you treat the mice with the antibody, you no longer are able to protect synapses. That'll be addressed in future work.
That's great. Have you seen this phenomenon in? I mean, I know maybe it's moot because you see this in HD patient brain, postmortem brain, but do you see that there's a CAG repeat length dependency in the complement upregulation that you see in mice? So does a mouse with 175 CAGs have more upregulation than a mouse with 60 or 90 CAGs?
It's an excellent question, and it's not something we've explored, but it's something that definitely should be addressed in the future.
Thank you, Daniel. So hopefully, I hope that with some really great questions, and I thank you, audience, for all of those really knowledgeable HD community. And I appreciate you dialing in. I also appreciate, Daniel, you taking the time out of your day to present this really exciting work. So thank you for that. And also, Daniel will also be, isn't that correct? Daniel, you'll also be joining us at the HDSA convention.
Yes, that's correct.
So we have a new session at convention highlighting some of our great work by our human biology fellows. So Daniel will be taking part in that session at convention in Baltimore. So hopefully you can join us there. And thank you again for joining us today on the HDSA Research Webinar series. So look forward to sharing when I have details of upcoming webinars, hopefully in April and in future months, take a look in your email box, and hopefully we'll be sending you updates as we confirm speakers for future webinars. So thank you again. Thank you, Daniel, and look forward to having everybody on the next call, hopefully in April. Thanks again.
Thank you.